Welcome back! Today, we will continue our journey exploring the fascinating world of evolutionary evidence and population genetics. During this discussion, we will practice distinguishing species from populations, analyzing allele frequencies and genetic variation, and exploring the Hardy-Weinberg equilibrium and its implications for evolution. By the end of this lesson, you should have a solid understanding of these topics and their significance in evolutionary biology. And with that, let's get started. Let's begin by defining two important terms: species and population. A species refers to a group of organisms that can interbreed and produce fertile offspring. It represents a distinct lineage with its unique characteristics. Members of the same species share a common gene pool and can exchange genetic material through reproduction. As an example, let's consider the American robin. American robins across different regions can mate with one another and produce fertile offspring. This interbreeding capability establishes them as a single species. So American robins from New York, California, or any other location can interbreed and produce viable offspring that are also capable of interbreeding. A population, on the other hand, refers to a group of individuals belonging to the same species that inhabit a specific geographic area and have the potential to interbreed. Populations may have distinct traits, though, or characteristics that are influenced by factors like geographic barriers, local environments, or genetic drift. As an example, let's focus on a population of American robins that reside in Central Park, New York City specifically. This population consists of all the American robins living within the boundaries of Central Park. They share a common habitat and have the potential to mate with one another and produce offspring. The Central Park population may have unique characteristics or adaptations specific to their environment, such as foraging behavior influenced by urban surroundings. So to summarize, a species represents a broader category of organisms capable of interbreeding, while a population is a subset of a species found in a particular geographic area. The example of American robins illustrates how they form a species by interbreeding across regions, while the Central Park population represents a subset with its distinct characteristics within that species. Individuals can belong to the same species, yet form distinct populations. This is a very important distinction to keep in mind when studying evolution and population genetics. In the context of evolution, studying populations allows us to explore how genetic variation is maintained and how it changes over time. Remember, evolution happens at the population level. Actually, as a brief check-in, what is the official definition of evolution? Right, it's the change in allele frequency in a population over time. So to explore evolution and characterize genetic variation within a population, we can look at allele frequencies. Alleles are alternative forms of a gene, and allele frequencies represent the relative abundance of each allele in a population. By analyzing allele frequencies, we can gain insights into the genetic diversity of a population. Alleles arise through mutations, or changes in the DNA sequence. And mutations introduce new alleles into a population, creating genetic variation. These variations serve as the raw material for natural selection and other evolutionary processes to act on. Now let's have a quick interactive activity. I'll provide you with an example. Imagine we have a population of 100 rabbits and a gene that controls coat color with two alleles, big A and little a. Among these 100 rabbits, 60 have the big A genotype, 30 have the big A genotype, and 10 have the little a genotype. let's calculate the allele frequencies for this population, starting with big A. Well, first, to calculate a frequency, we must know the total number of alleles we are dealing with, right? Just as a brief math refresher, frequencies are calculated by dividing a part over a whole, and are always between 0 and 1. Right, so we were told that there are 100 rabbits, and each rabbit has 2 alleles, right? Either big A, big A, or little a. That means that the total number of alleles in the population is 200, because 100 times 2 is 200. Does that make sense? Okay, so starting with calculating the allele frequency of big A, we are told that 60 rabbits have the genotype AA, so since there are 2 large A's in this genotype, we multiply 60 by 2, which is 120 big A alleles from these rabbits. We were also told that 30 individuals have the genotype big A, little a. Only one big A in this genotype, so we would add 30 to our previously calculated 120, which totals 150. So our allele frequency for big A would be 150 divided by 200, or 0.75, right? Yay! Okay, so that's the allele frequency for big A, 0.75. Now it's time for little a. There are 2 ways we could calculate this. The first way is to take the presented information from the prompt, calculate the frequency that way, like you can see on the screen here. The second, shorter way though, would be to take your frequency for big A and subtract it from 1 to get the frequency of little a, like so. Remember, we are dealing with frequencies, so adding both parts of the whole together should always equal 1. Now, regardless of the way you do it, you should get 0.25 as the frequency of the little a allele. These values mean that 75% of the alleles in this rabbit population for coat color are big A, while 25% of the alleles in this population are little a. This information gives us a snapshot of the genetic variation in the population. All right, now, as your next checking question, you give it a try with the scenario presented on your screen. Feel free to pause before moving on. Keep these practice calculations handy, as we are going to revisit them later in this lesson to help determine whether or not a population is experiencing evolution. Okay? In the meantime, though, now that we understand allele frequencies, let's explore the sources of genetic variation in a population. Genetic variation arises from several processes. One primary source is mutation, which introduces new alleles into a population. Additionally, recombination during the formation of gametes during meiosis, sexual reproduction, as well as gene flow, which is the movement of genes between populations due to immigration or emigration. All of these also all contribute to genetic variation. It's important to note that genetic variation provides the raw material for natural selection, the driving force of evolution. Individuals with advantageous traits are more likely to survive and reproduce, leading to changes in allele frequencies in a population over time. So, genetic variation is the diversity of alleles within a population. We can measure this variation using allele frequencies, which represent the relative abundance of different alleles in a population. Now, let's delve into the Hardy-Weinberg equilibrium.